But now the first Plant Phenome Journal monthly webinar was 2 years ago, I think today approximately, or at least in the month of October. So I think we've had about 20 webinars since then. So my name's Seth Murray. I'm professor at Texas A&M University and the editor at the Plant Phenome Journal who handled our speaker's article. And so I'll be moderating the discussion and helping facilitate this. So Dr. Garett Heineck, I hope I pronounced that correctly. I should have.
Correct. Yep.
Okay. Dr. Garett Heineck is a recent PhD graduate of the Applied Plant Science program at the University of Minnesota, and he's focused on plant breeding and genetics. He completed his bachelor's at University of Wisconsin River Falls, and currently he's a postdoc working on turfgrass seed production systems and intermediate wheatgrass. He recently published a paper in the Plant Phenome Journal on using R-based image analysis to quantify rust on perennial ryegrass, and that's what he'll be speaking on today. And please consider your questions. You can type them in the chat, and if you have any problems, type it in the chat. And then after he concludes his presentation, we'll open it up for questions either through the chat or through open microphones for those people who want to ask questions. So without any further ado, Garett, please, we look forward to your presentation. Go ahead and get started.
Oh, thanks, Seth. Yes, today I'll be talking about using R-based image analysis to quantify turfgrass traits. And a large portion of this presentation will be based on the recent publication in the Plant Phenome Journal. Yes, so as Seth said, my name is Garett Heineck. I'm from the University of Minnesota. And an overview of today's talk is going to start off with an introduction to turf in general. I don't know how many turf scientists we have listening in, but I imagine there will be some folks who don't have a strong turf background. So I'm just going to start off just with kind of how we conduct our science on turfgrass, especially turfgrass breeding, and what different turfgrasses are used for. Then I'll get into image analysis to quantify how you can use image analysis to quantify rust on turf. And then some other image analysis techniques I've been working on to quantify turfgrass traits. So if you think about the importance of turf, I like to show this figure that shows kind of the hot spots of where turf grass is often grown. And as you might expect, oftentimes it's centered around large metropolitan areas. For example, I'm from Twin Cities, right in Minnesota. You can see a large green dot right where the Twin Cities are. So where people gather and are clustered at high densities, oftentimes we see high densities of turf right along with that. So they're an important part of many of our lives from an aesthetic standpoint, as well as some other aspects as far as storing carbon to a certain extent and remediating water and things like that. So oftentimes when we think of turf, we think of it as a lawn, right? Either a home lawn, or maybe it's outside of the building where you work. But turfgrass is also important for sports fields. Here's a picture of a sports field that's probably got some perennial ryegrass growing on it, or some other species that works well on sports fields. And our lab in particular does research on turf grasses. And one thing that we focus on is sports fields. I won't be speaking a lot about this, but I think it's an interesting eye-catching figure here by one of my lab mates who he tracks sports field or sport players' movements and figures out where they get hurt, and then correlates that with different parts of turfgrass quality traits such as density or hardness. So oftentimes, if we can see on the figure on the right where the black stars are, oftentimes where players get hurt, which is a black star, is on the periphery of these compacted zones, because oftentimes players are trying to move around them. So there's a lot of interesting research that can be done on turfgrass. Also, one place where people don't really think about turf being important is on the roadside, on roadsides. So here's a picture of a roadside in a town that's got some turfgrass. And our research lab does a lot of research on roadsides, specifically what turfgrass cultivars or species or mixtures of species and cultivars work well on roadsides. So here you can see along this road, there are like little patches of turf. And if any of you are familiar with turf research, you won't be surprised by that. But if you're not, this is how we often conduct trials. We make these little patches and we measure how well the grass grows within these little plots. And this one just happens to be alongside of a road. For breeding work at the University of Minnesota, we've got a fairly large breeding program. This is a picture of one of our breeding trials. We just took this using a little UAV. And you can see all the little, like the little checkerboard pattern and how the different cultivars look differently from one another. And this is simply how we screen our turfgrass by looking at different aspects of the turfgrass quality. If you think about our breeding program in general, I'd just like to, since I am a plant breeder, I'm going to go over really quickly how we conduct our programs. And I'll start off with, in the top left-hand corner, the selection nursery. Oftentimes we start off by selecting just individual spaced plants. From there, we might look at our favorite space plants, the seed that's produced from them in turfgrass plots for a couple years. From there, we might take those, our favorite selections, maybe this plot here, take the parents of those, of that population and polycross them and send it on for, to crossing blocks to increase the seed for production or maybe increase testing further. One thing to note is here's a field of spaced plants. When I'm saying spaced plants, so the plants are spaced apart and we're phenotyping them individually. So we can, as turf breeders, we're either phenotyping a plant or a plot. And oftentimes in today's talk, I'll be talking about spaced plants. So what did the plant look like, or what did the leaf of the plant look like as far as disease severity or some other trait? And this is just what I'm talking about. We screen several thousands of these. Another thing to consider is from a turfgrass plot standpoint, we often have multi-location testing happening. So after we get done with our years 2 through 3 of selections in our own turf plots on site, we might polycross and send our favorite selections out to maybe New Jersey or Wisconsin or California. And that's done actually through the National Turfgrass Evaluation Program, or NTEP handles all this data. So NTEP really, I was just talking to Seth about this, it takes all of our university produced and industry produced cultivars and tests them side by side. And so in the guidelines for NTEP, we, for one, rate turf quality. That's on a 1 to 9 scale, and genetic color on a 1 to 9 scale, and turf density on a 1 to 9 scale, and percent living ground cover on the 1 to 9 scale, and on, right? Texture. So everything's on a 1 to 9 scale. And as you can imagine, even if we use color palettes like this one to try to rate color, or one like this to rate density, oftentimes we get different raters from different parts of the country rating differently. And if you look at the national results, our lab is working on kind of synthesizing all of the results into an easily understandable format for a mobile app. You can oftentimes see that different raters at different universities have higher or lower expectations of what turf quality is. And so I think this is an interesting example of where we can use image analysis. Just to show a little bit more, exemplify this a little bit better. If we look at this turf plot here at the top of the screen, on a 1 to 9 scale, 9 being the best and 1 being the worst, you could imagine this being quite a few different numbers, right? Maybe it's an 8, maybe it looks really good to you. Maybe it looks like a 3, maybe compared to your lawn at home, it looks very poor. But what really sets the actual number is comparing it to something else out in the field. So what a rater rates is always relative to what else is in the field, because that's what you see. And so for an example, something cultivar A that was measured in 1960 might have been a 9. But compared to new elite cultivars in 2019, it cannot be a 9 anymore, or else you cannot make genetic gain, right? So these skills are very difficult to use from that standpoint. And that's one thing that I'll be talking about today is how can we use image analysis to better quantify these traits that are so pervasive in the turf field. If you think about percent living ground cover, it's another fraction of the turf quality index. And ground cover itself can be measured several different ways. So we can have insect damage. Here in the lower picture, we see some damage from white grubs, weed encroachment, environmental stress. In Minnesota, winter kill is especially difficult to deal with from a breeding perspective, very hard trait to breed for, low heritability. and disease damage. So crown rust, and that's the first thing I'm going to focus on today. So oftentimes crown rust for turf breeders is measured on, again, a 1 to 9 scale. I'll be talking about the modified Cobb scale, so maybe a 1 to 100 scale or 0 to 100% scale, but it's still subjective, right? And I just like this quote from Sherwood et al. 1983. So he's talking about trying to train raters to rate for a disease in forages. So training alone will not eliminate illusion. We are presently evaluating computerized analysis of video images as a potential system. And I just, as soon as I read this, it really resonated with me because I have the same problems. I'm also a big Star Trek fan, so I automatically thought of like Leonard Nimoy and the laser disc, Magnavision. So that's my shameless Star Trek reference there. But moving on, image analysis in Turfgrass should be relatively straightforward. I've watched several of the webinars on YouTube, and I think that compared to what a lot of people have to deal with, turf is pretty easy. So we're mostly dealing with aesthetic traits. So oftentimes what our eyes see is what we're measuring. So what RGB cameras can pick up should do the trick in many cases. They're mowed regularly, right? So we're dealing with a 2D, could be thought of as a 2D surface here. It's always 2.5 or 3 inches or some combination of that, depending on what you're doing. But it's not like a corn plant that's allowed to grow as tall as it's going to get. And if you take a picture of the top of that, right, you don't know how many leaves are underneath that. So you're not dealing with leaf area index. It's a very 2D game for analysis, which is nice. So we should be investing more in image analysis is kind of my point. Some commercial and open source software platforms that I know people have dabbled with, Assess for diseases, Canopeo, which is a nice open source and it's got a mobile interface. And then Turf Analyzer is becoming more and more popular in the turf realm, but it's also a commercial product. Works really well though. So if you are turf scientists out there, I'm sure maybe you've heard of this or have used it for analyzing your turf data. But today what I'd like to talk about more is open source. Here we have ImageJ and Python and R, which are all open source platforms or languages that we can use to analyze images. And I chose R in my lab instead of ImageJ, for example. It's because it's such a popular data science language. For example, graduate students in statistics are, at least at the U of M, are always introduced to R for data analytics. And then I help teach applied experimental design, and over 80% of students enrolled in that over the years have used R. So I think analyzing images in R if possible, and then using that same data generated in R to do analyses in R is not a bad way to start. So that's kind of where I went. And with that, I'll start discussing my recent publication in the Plant Phenome Journal, how we can use this R-based image analysis to quantify rusts on perennial ryegrass. As a quick aside too, perennial ryegrass is common turf grass used for sports fields, home lawns. You probably seeded in your lawn at some point in time. So crown rust reduces the aesthetics of both turf and forage swards, especially perennial ryegrass. You can see here, I just have a picture on the left taken 7/24/17, and then just a month later, crown rust has completely destroyed that nice healthy green tissue. So one thing we're often interested in is rating crown rust. Here's a scale that I use. Typically, it's the Modified Cobb scale from Rolf's 1992. And you can see here's a diagrammatic scale that we can follow, right? We can train our eyes on this scale and compare it to what we see on a leaf, and then kind of extrapolate that to a whole plant or to a plot if we so choose. So you could think of a leaf looking like 50 here, and then writing the 50 down and hoping that leaf is 50% infected. So here's just showing that. So maybe this leaf here is 30. But as far as raterability goes, based off Sherwood et al. And many other publications in plant pathology literature, we know that raters often have low precision, right? From one rating to the next, from one rater to the next. oftentimes precision is low. Overestimation is common. So if we look at this top image here, oftentimes what a rater will see is actually at the bottom here. If you think about, it's kind of like interesting, there's some interesting papers almost kind of getting into like psychology of rating, where we tend to ignore healthy and focus on disease. And then raters oftentimes have a hard time distinguishing different diseases and then counting the number and size of the disease entities. So here we're looking at pustules of stem rust here with the darker red, burnt orange type arrow, and then crown rust on the right with the lighter arrow. And so as a rater rating this image, I'm asking myself to rate both all the stem rust independently of the crown rust, and that can be difficult to do. So these are all things that I think image analysis could do a better job of than a human, or at least as good of a job. So questions that I had for this study was, what is the consistency and agreement between rater scores? Right, so how can we trust rater scores from one rater to the next? What are the perceptions of different raters for diseases? And then, can the median score be used as a validation for image analysis? Meaning that, can we trust what a rater thinks is, say, a 50%? Is that actually a 50%? So these are two questions that I sought to answer before I'll get into the image analysis itself. So the experimental design, I just took 50 plants for this study. There's a larger validation set that I'll get into that I used as well. But at first we did 50 plants. They varied in disease resistance, and there were 3 series for each of the plants. There's a whole plant image, and then I sampled 5 leaves from that plant and imaged those, and then I randomly selected 1 leaf of those 5 leaves for the 3rd image series. So it's kind of this almost like a little nested design, right? We've got our whole plant, and you could think of these 5 leaves as representing that whole plant to a certain extent. And then the most objective view that we have of a plant is a single leaf. Oftentimes we use, in the turf world, we use light boxes to capture images. And so in this study, I either use a light box or I use ambient data, but here's an example of a light box that we could use. This is just I just made this. It's like a chloroplast sided with lights on the interior with a 12-volt battery that supplies power. Here's a smaller one that was used for the leaf, 5 leaf samples, and that worked very nicely to get, it gives you really consistent lighting. Simple RGB images taken with a Nikon D300, nothing fancy here. Anyone can pick this up at National Camera for like $100. So really simple technology being used in this study. To test the ability of raters, I chose 9 raters from 3 different departments to rate each of these series. So we had raters who were either from experts on Rust to folks who they didn't, never even hardly rated Rust before, but they have a lot of experience rating maybe other diseases or using other visual scales. They're all scientists. And then I trained them on a set of calibration images, and then they each independently rated each of these 3 series for crown and stem rust. And if we look at the intra-class correlation coefficients that resulted from this, which was set to calculate consistency, meaning what maybe rater A thought was good, rater B also thought was good, and what rater A thought was bad, rater B thought was bad, right? So we're not talking about necessarily absolute agreement, but in general, the consistency of our scores. And for crown rust, people had across image series. So we have in gray whole plants, and then moving over 5 leaf and single leaf samples. All of those were very excellent ICC values, where stem rust was slightly lower. And what this tells us is that certainly for crown rust, people had high consistency of what they thought, what they perceived a bad or good plant to be, bad or good leaf to be for rust and severity. Coefficients of variation across the data sets. or quite high, meaning that the actual values that people were scoring of the image series were very variable. So at best, we had a CV of 50%, which is quite high. Stem rust was even worse, and that'll be a trend throughout, even in the image analysis, is stem rust was just harder to rate, especially on whole plants. So larger CVs, higher variability. And this we found was very interesting. If we look at the variance around the median score, the agreement, right? What were people's perceptions? If we look at the panel on the left for crown rust, and we just look at single leaves, the single leaf is where we would expect the highest level of precision to be, right, for raters. But we saw tons of variability there. And so what this told us was that hopefully the median score is quite accurate, but certainly between raters, we had a lot of variability. It's quite interesting to see. And then rater accuracy between the series, if you look at the bold numbers here, the top bold number, rater scores between 5-leaf and whole plants were very high. This indicates that we had a high level of accuracy. And then for 5-leaf, the whole plant series for stem rust were quite a bit lower, meaning a little bit lower accuracy. So in conclusion to what we found with the raters is we found moderate to high consistency for crown rust especially, but poor agreement, fairly poor agreement across the board. However, we used median rater score for validation, but we still move forward with some caution, especially as far as stem rust scores go. So for the image analysis system, finally, we used the visual field ratings, the manual measurements, and ImageJ to validate our computer measurements. And we used R language and ImageJ to generate training data. So it's these two open source programs. And the first step in the process that I use is creating training data. So teaching the computer what is what. So if the person using the system doesn't know what they're doing, right? So here's a leaf full of crown rust. It'd be rather difficult to teach the computer what to find. So make sure that for crown rust, you know what's crown rust and what's stem rust, and that's the best way to proceed from there. So the system relies upon creating mixes of different RGB profiles in ImageJ. And if you go on my GitHub page, I have a tutorial on how to make these. It's really, really simple. In fact, there might be better ways, and if someone has a better idea, to certainly let me know. The mixes are then combined into a training palette. And the palette is kind of a collection of RGB profiles that helps you define an object that you want to isolate. So I like to think of it as like, if I'm Bob Ross and I'm trying to paint a tree, I'm going to be using a lot more like forest green than titanium white, right? I need to define my object and then figure out what color profiles, especially from a turfgrass standpoint, right? It's all about aesthetics. What color profiles match that object that I'm trying to isolate. And here's an actual demonstration of this, where data points 1, 2, and 3, if I were to click on those spots in ImageJ and record those RGB profiles, that would be for, let's say, a healthy leaf, or on the periphery of the stem rust pustule, where 4, 5, and 6 are the actual, it's like the sign of the disease. It's the actual pustule, and the rust, we are very lucky to have the sign of the disease present. So 1, 2, 3, healthy, 4, 5, 6, infected. And then we use the training data, right? These different mixes to create a random forest model. There are many different functions within, like, say, the Garett package that you can use. We found random forest across several diseases seem to work just fine. Maybe there's, for your system, a different algorithm will work better. So check out other ones in the Garett package, but random forest works really well for us. So that's what we used. And then we also, after we applied this random forest model to our image, we then used EB_image to do further processing. So here's just kind of to display or portray what I'm talking about. A is the original image of a leaf, and then B is using random forest to pull out the leaf itself and remove background, so you have foreground and background mask, and then you can reapply those original RGBs values to the foreground. As far as what EB image can do, it's actually really, I think it's used for mostly cell counting and things, but of course you can use this for plants too. I think maybe Plant CV and Python might have a little bit better functionality or a little bit different functionality maybe, but I found EB image to work just fine. So here in this example, we see that we've got many, many small pustules all clustered together in the top image. And the random forest model is having a hard time defining where those pustules are because the color profiles are so similar. But we can apply a watershed transformation to look for peaks and valleys in that mask and get a not a perfect definition, because I don't think even our eyes can get a perfect definition of how many pustules are, but get a much closer idea of how many rust pustules there are and what the severity truly is. So again, the computer accuracy was validated against the same 3 image series that the raters used. And then I also validated it on the modified Cobb scale itself. As you might expect, it worked really well, almost perfect R squared, so it can predict the Cobb scale. But a more challenging thing, of course, is to find out if it can actually predict pustule numbers on the single leaves. So as far as crown rust went, we had 84% variance explained for the number of pustules, and then 75% variance explained in stem rust. Stem rust is just typically a little bit more difficult to deal with. As far as total infection of rust, we had 92% variance explained on these single leaves. Again, the single leaves are the most simple of the 3 series, but it did an adequate job of explaining severity. And the severities were actually manually calculated. So this validation set was actually against true severity, where I actually went in, we had several people go in and actually trace around each pustule to calculate severity. And then we actually also validated this on a larger data set. So we went in and correlated the computer scores versus visual scores on a larger data set of about 560 across 2 years. And we found very similar 78% variance explained and 74% variance explained. So that validation was fairly positive. But then how did these, to really answer the question of how did these, within our rater panel, how did the computer match up, or actually never mind, sorry, this is still between different series. So similar to visual rating, crown rust had very high accuracy, where the 5-leaf sample correlated very strongly with the whole plant severity. And then stem rust, we had a very low correlation, just similar to what the raters had. Stem rust was difficult to rate for the computer as well. Oh, and then now we get to how does the computer relate to the rater? So the computer score versus the median rater score. For crown rust, across all 3 series, we had fantastic correlations, all above 0.9 for Pearson correlation coefficients. For stem rust, unfortunately, we've had a non-significant correlation between what the rater thought was stem rust and what the computer thought was stem rust. We didn't have a good correlation there, but for 5-leaf samples, it did work fairly well. We'd have a significant correlation there. So again, stem rust is a little bit more difficult to deal with. And then for stem rust, we looked at, well, maybe the computer is more accurate than the person, or it's rating something differently than a human rater is for stem rust. And if we look at all 9 raters, and then you have an experience level in the second column, 1 being low experience and 3 being an expert in the field, we see that really there's no trends between what the raters thought had a lot of stem rust and what the computer thought was stem rust. So in the end of the day, what we really thought, what we concluded was both the computers and the raters were measuring totally different things for stem rust. But for crown rust, it worked quite well. If we look at actually, can we deploy this system in the field? I wanted to look at data collection efficiency. And what I found was that it took me on average 6 seconds to rate a plant for crown or stem rust. An image took about 4 seconds. and then of a whole plant. And then an image of 5 leaves took about 140 seconds. So we saw between the 2 image series, a huge difference in the amount of time taken. So really what this says to me is very few people are going to take the time, especially in a breeding program, to measure a 5-leaf sample. It just takes too long. But a whole plant sample or whole plant image is quite simple to take, only 4 seconds. Probably very similar to a visual rating. If the system does work, and it seems to work for crown rust, I think in our program at least, we're going to be switching over to using more automated analysis. But just to give the viewers, this is kind of a fun demo of how the system ended up working. If everyone looks on the scale here, this is the Modified Cobb scale, and you look at the leaf, try to assign in your mind, this is like a little game, what panel matches that leaf for total rust infection. Or you can do crown and stem rust independently and add them together, I don't care. Just like look at which panel matches the leaf and then remember that number. Was it a 5, was it a 50, was it a 40? And then I'll switch over to the next slide here. Our rater panel measured this leaf at a 30 on the Cobb scale, which translates to actually 11.1% total severity as far as the leaf coverage. Manual severity calculation put this actually at 3.5% severity. So 3.5% versus 11.1%. And this is very typical to what people have been finding since the 80s or before, that raters typically overestimate, right? We focus on the disease rather than the healthy tissue. And then the computer, this system in particular, this is a fairly good example, but found 0.4% stem rust and 3% crown rust for a total of 3.4% total severity. So in this case, and in many cases, the computer was much closer to the actual percent severity than the actual rater panel was, especially for single leaves. For whole plants, it can be kind of tricky, but in this example, kind of just, for me, what it kind of shows that sometimes raters overestimate what severity was, and they do in the field as well. So in conclusion, inter-rater reliability scores showed considerable consistency, but we did see low agreement, meaning what one person thought was a 10, someone else might have thought was a 30. The automated system that uses R and ImageJ was fairly pragmatic for crown rust rating in particular, and was quite accurate. And I think it's probably ready to be deployed, at least in our breeding program. However, neither the rater nor the computer was adept at rating stem rust on whole plants. So if we wanted to at least using this system, in the paper I have several ideas on how we can make this better. I don't think we should give up on it. Certainly there's a lot better cameras you could use, or better analysis strategies, breaking down the image in different ways that could be applied. But this particular system, when I have on GitHub, does not work very well for stem rust. And now I think I have time. It's only 11:34. I have time for some bonus features. So different ideas that I have for applications for these simple image analysis techniques for turfgrass quality traits. So on my GitHub page, I'll show this at the end, but I've got a tutorial for whole plants and leaf samples for stem and crown rust. So please feel free to check that out. If something doesn't make sense or you think I could do something better, I'm really open to suggestions. I'm not a computer programmer. I'm a plant breeder trying to kind of bridge the gap between the computer science folks who I work with, and then the plant breeders and agronomists in my lab who I also work with, and kind of trying to make a system that is simple enough so that someone coming out of like an intro to stats course could pick up a script and apply it to some images with some training data and get something to come out that gives some meaningful information. So moving on, from a turfgrass breeding program standpoint, these are experimental units. Each little square of turf is an experimental unit. We want to measure that. So here we have about 450 plots. That's a fairly, at least in our program, a fairly typical size of a trial. And NTEP would have us go out there, and this is a system that works very well, go out there and rate turfgrass quality and genetic color and on a 1 to 9 scale and report back to them. And it's a system that's driven gains in the industry. but I think maybe we can do it a little bit better. So thinking about one problem that we have in Minnesota and many places in the upper Midwest is winter kill. So here's a plant that suffered major winter kill, fairly severe, and we can see some of the part of the plant is alive, some of it's dead, and then there's soil around it. Other complications were a lot of algae actually in the ground, which is fairly similar to what the plant itself looks like. So what is winter survival? Well, it's the proportion of a leaf tissue that's alive versus the proportion of leaf tissue that is dead, right? It's not just how much is alive within this image here, because different plants are different sizes. So if we just looked at the number of green pixels, we would not get accurate information on what actually happened to this particular plant. So first, to remove the noise that occurred because of that algae, I just created a mask that really blurred the image, and then all the high probability pixels from the random forest show up, and then I overlaid the original RGB values from the first image that I showed onto that, and we can remove kind of the background noise. It's not perfect yet, right? But now we can apply another round of random forest, a more picky, I guess, random forest model to this middle bottom picture to extract out all the green leaves and then subtract that from all the dead leaves. And that gives us winter survival. Here's another example of winter survival. You can see in this one, just a few little tiny leaves that are alive on the bottom of this plant. It's really, really pretty much dead. We can apply the same principle and then again, pick out on the picture on the bottom right, a few little living leaves. Compared with visual scores, we've found this to be very useful. R-squares at 2 different locations. One R-squared was predicting, or looks like we had R-squared of 91% variance explained and then 85% variance explained at location 2. So this has been a pretty fun thing to work on in particular. Here are 3 different turfgrass plots. These are imaged using a light box like I showed before. So we have consistent lighting across all 3. And actually, in the beginning of the talk, I showed you plots 3, and then I showed you plot 2. And so I had just said at the beginning, it's difficult to compare different turf plots on a 1 to 9 scale, which we typically use, if you're not comparing them from one to the other. And so I had our breeder, or my advisor at the U of M, rate these, and he graded number 1 as a 2, number 2 as a 7, number 3 as a 4. And so it's easy to see why he said that, right? Plot 1 has got a lot of crabgrass, it's got a lot of stems, it's got a lot of problems. Plot 2 is fairly good, it's a 7, but maybe compared to something that's really a lot better, maybe it wouldn't be a 7. And plot 3 was rated as a 4, okay? But why were each of these rated this way? If you saw this data on NTEP, you would have no idea why plot 1 was a 2. Right? You don't know that there's crabgrass, and then you don't know how that it was stemmy. And even if you did know that there was crabgrass, you wouldn't know the exact amount. So these are things that I think image analysis can help with. So it's a simple example here. So let's first look at green cover. You can see that this is, I whipped this up, it's fairly simple to do. The bottom is showing all the green, healthy green tissue that I just defined as green tissue anyways in the model. It pulls that out, and we see that plot 1, of course, has less green tissue, healthy green tissue than plot 2, but actually slightly more than plot 3. So really, plot number 1 that was rated very poorly, it's not doing that bad for the number of healthy leaves as far as like the proportion of total turf area. Then stems. Stemminess is the residual of when the plant is producing seed heads and we mow them off and we keep mowing them off and then they dry out and looks like straw. The image analysis system can pull those out as well. And we find that if we quantify that, again, plot 2, which was a very nice looking plot, had very low proportion of stems. Plot 1, which was very bad, actually wasn't even that bad for stemminess. And then moving on to crabgrass, we can pull the crabgrass leaves out. Plot 3 here, this just shows that it wasn't, the system's not perfect by any means for, this is a preliminary thing I'm working on. it's not perfect for crabgrass. It's definitely, I'll need a different system to figure this out rather than just simple color values. But you can see for plot 1, it does pull out those crabgrass leaves fairly well. And we can quantify that. And lo and behold, plot 1 has many, many, many times higher crabgrass infestation than plots 2 and 3. And so if we combine all this data together, where I hope our industry is going is using this data routinely and uploading it to either using it in research papers or uploading it to the NTEP website. And then when we see turfgrass quality ratings of a 2, a 7, and a 4, we can look at other data and we can say, well, why was that a 2? Well, it's really not because it didn't have a lot of healthy leaves, proportionally healthy leaves, or too many stems proportionally again, but really it's just because it had a lot of crabgrass. So if that's a problem that you're concerned with, then you would be able to take that information and use it accordingly. So again, my GitHub repository is just Garett Heineck, all one word, and then this little QR code that takes you to the website as well. I just have 5 tutorials so far. Feel free to check them out or email me at my UMN account, or my GitHub email address is fine too. So I hope you learned a little bit about turfgrass at the University of Minnesota and how we do national trials, and hopefully a little bit about how I think image analysis is gonna help us in this field in the future. So thanks everyone for listening. I think that's about my 40 minutes. So thank you.
All right, well, thank you, Garett. So we're gonna open it up for the next 15 minutes or so for questions. So you can either write that in the chat box, and I haven't seen anybody other than me write anything in the chat, so I hope it's working. Or if you'd like, you can unmute your microphone and go ahead and ask Garett the question. While you're preparing for this, I'll just make a plug for the Plant Phenome Journal. I mean, I think this was a great example of what we're trying to promote. A lot of sort of new imaging analysis techniques for helping breeders or helping field scientists make decisions. Great article, and excited that we could publish it.
Thanks.
So, Garett, I'll start off with a question here while we're waiting to see if anybody else has a few questions. So, from a plant breeding perspective, you talked about raters were inconsistent. I think a lot of what you were focused on was this move to objective data. But does the consistency of raters in terms of comparing their absolute score matter as much as consistency of rankings between the raters? So to select the best variety, regardless of, maybe one rater thinks all varieties are poor and another one thinks they're all great, so the consistency would be low, but the rankings might still stay the same.
Right, that's a great question and something that I've thought a lot about. I think it doesn't really matter in certain circumstances, especially when you only have one rater rating your plots or rating your plants for disease, for instance. But when you start comparing ratings across institutions, or when you have maybe my undergraduate goes out and rates, and then maybe I go out and rate, and then maybe my advisor goes out and rates. When you start trying to analyze your data across raters, that's, I think, when you start running into some problems, especially when you start generating like AUDPC curves and things, because you might be, although your rankings may be very similar, the actual maybe like the marginal mean of a certain cultivar is going to be off, just because you've got multiple ratings. So I think if you're really diligent and you are the only one rating your turf plots, then you're probably okay. But if you have multiple raters or multiple institutions doing ratings, this is when we start to get into trouble, especially with NTEP data, I think. So does that answer your question, or?
Oh, absolutely.
Okay.
No, I think that's great points. Would anybody else like to unmute? Because I'm not seeing anything in chat.
Garett, this is Ambika Chandra. I am a turfgrass breeder located here in Dallas Center. Great presentation, enjoyed it. And one of the things, this is more of a comment than a question, is your latter part of your presentation where you talked about identifying why something is getting a low or a high rating. That is very important. When we get NTEP data and we see a certain variety is doing poorly or really well in a certain location, it's rated as winter kill or it's rated at something, and it's hard to dissect why it is rated poorly or well. And so how do we go about, doing it, being able to do it at a certain location where we have experts like you to spend the time and dissect, tease that out, versus developing, maybe going to a point where we can develop an algorithm that is incorporated into these programs that helps us to dissect, find details of what we are seeing and why we are seeing what we are seeing.
Oh, that's a great point. I think this is not necessarily my vision, but I think what Eric Watkins, my advisor's vision at the U of M is to take the NTEP data, right? It's a big data set, right? And it could be a lot bigger. But in order to utilize it, right, we need to have it in a format. Right now, it's just in a bunch of text files. We need to have it in a format that's queryable, from probably using SQL or some other language, extracting data from that, and then being able to, from a very basic consumer standpoint, just synthesizing those results into even means. But from a research standpoint, we could be using that to validate, right, to your point, to validate image analysis systems. And so that instead of being like, oh, yep, this cultivar did really bad at Rutgers, but it did really good at Minnesota, we could query this maybe for very specific responses and have it be on the same scale. Because if the image data is all taken using the same or very similar systems, then we should be able to discover a lot more about those cultivars' performances across locations.
Absolutely, especially across locations. I have dealt with data where you get two locations that are not very far from each other, and then latitude-wise, they're pretty same, and you get data on winter kill, which can be very different.
Oh, absolutely.
And how do you make sense of that in terms of especially winter kill? Why are you getting more winter kill in one location versus the other, given that they both are in a similar latitude? And so there's clearly a lot of value in finding out why, answering that why question. So really, really nice work.
Yeah,
thank you.
On top of that, winters, thank you so much for that, that comment and question. If you look at the screen, the picture that I have up right now, in the lower left hand, you can see winter kill on perennial ryegrass. And this is not winter kill because the genetics in that corner are poorer than the genetics anywhere else in the field. The winter kill is simply worse there because that's a poor spot in the field. And in fact, you can see diagonally across this picture, you can see actually footprints. We had people walking through this this particular plot. So winter kill especially is so spatially messy that having image data with the coordinates of where the plots were in relation to each other and doing spatial analysis, spatial statistics would be just fantastic. I think that's would be a great next step, especially in breeding for winter survival and winter hardiness.
Absolutely.
Thank you.
Garrett, I'm glad you brought that up, because that was my my next question, if nobody else had one, was field spatial variation, because I was actually expecting that the bottom left-hand corner in this picture is where people were entering and exiting the field. And so have you done any work to try and adjust for that?
No, not yet. It's on the agenda, especially now that we're getting these, we're starting to get more into the taking data via drones. And of course you wouldn't need to take it. You could just use simple light boxes, and you could make a spatial point process model based off XY coordinates of your data. But that's something that we are very interested in working with, and something that we're trying to collaborate with on the winter grains, with the winter grains folks, winter wheat, winter barley, to see how they kind of set their models up for predicting what an actual winter survival score might be, right? Because obviously in this picture here, I like it because that has nothing to do with genetics. So it's almost like a nearest neighbor type of approach, where if your neighbor is heavily winter killed and another neighbor is heavily winter killed, well, that's probably just an autocorrelative structure where the plot, there's like an epicenter of death, and it kind of spreads out in an autocorrelation fashion where 10 plots away, there might be no winter kill or less winter kill, but close to that epicenter, you have high winter kill. And this kind of stuff makes NTEP data very difficult to deal with, especially because winter survival drives turf quality, can drive turf quality for 2 or 3 years, because these plots are never going to look good in the bottom corner. They're just going to be terrible. So those kind of spatial adjustments, I don't have any particular plans for, but I do have ideas on how might be a good way to move forward on that. So do you have any ideas, Seth?
Well, so for my program, of course, we're looking at yield, so it's always a little different than turfgrass. But because we irrigate down rows that have been furrow irrigation down rows and fertilized down rows, we typically fit a row column factors in the model, and it typically explains 10 to 15% of our total experimental variation. So it helps to control for that. And as I'm looking at your image here, I mean, that front row, the one at the bottom, it only works if you have replicated plots. So if you have replicated plots, you would see that the one on that bottom row is heavily damaged, right? So that would help adjust all those plots up compared to, not accounting for spatial variation. And similarly, the third column from the left looks like it's similarly sort of disrupted. So just fitting it's really easy for people, especially when you got these rectangular field layouts, to fit row column effects and just see if it explains any variation. That's
a really good idea. Have you done any work with, or maybe someone in the audience done any work with, I guess maybe using augmented designs or some other system where you use like more of a nearest neighbor approach? I've heard of some people doing that, like the small grains people at the U are kind of doing that to adjust, not like the row column idea is fantastic. I'll probably look into that, but like from almost like a moving window standpoint, if anyone's used any of that kind of approach. But that's fine too if you haven't.
Yeah, I certainly don't. I mean, I tried fitting that when I first started my career, and the hassle of fitting it, and you can get a lot of other sort of weird effects. Whereas row column is, especially with a large field like you're showing here, is more robust. But it also is somewhat of an academic exercise at some point, because your goal is really just to make the best decisions, and you have to do it quickly. At least in a row crop breeder's case, you have to do it quickly. So
yes, absolutely, yeah. Yep, I think that's right on. That's quickly and consistently. So
yep.
All right.
Any other questions? I still haven't seen anything in the group chat, so I hope it's working. But would anybody else like to unmute and ask our speaker a question? Okay, I'm not seeing anything, so I guess with that we will conclude the webinar today. Thank you, Dr. Heineck. Very nice speaking to you. Great presentation and great paper. Look forward to other great things from you in the future.
All right. Well, thank you so much for having me on the webinar series.
Yep.